Alright, so today we're talking about gene expression. Now, one really interesting thing in biology is that all the cells from the same organism contain the same genome, the same bulk of DNA. However, they're not going to transcribe all of it to create proteins for every single one of the genes. In fact, in any one of our cells, we may only be expressing 10,000 genes of the 20 or so thousand that we actually have. Now, part of this is because of differentiation. And in all of our cells, so for example, think of a liver cell, and then think of a neuron, and then think of a white blood cell. All these are extremely different in shape and in function, but they all have the same genome, even though they're totally different cells. So how does this come about? It's basically all about how they are going to make and accumulate different RNA and proteins. So this is all part of gene expression in those different cells and how they're being expressed differently. So what regulates that? A lot of it's going to be on the actual level of initiating transcription. So we'll talk about that in a little bit. Some proteins, though, are going to be transcribed and produced in all cells. So some proteins are actually housekeeping proteins that are necessary no matter what cell it is, such as RNA polymerase. You're pretty much going to need that one in all of your cells. However, there's a lot of proteins in RNA that are specialized and will only appear in certain cells based on what the cell has to do or how it functions. All right, so moving on. Some of these things that will turn genes on and off are going to be extracellular cues, meaning they're coming from outside of the cell. And a lot of cells that are different can respond differently to the same signal. So our fat cells and our liver cells respond differently based on this glucocorticoid hormone. Our liver cells, for example, will release glucose, and now that they no longer need to store it for later on. Now, a lot of this regulation is going to be at the level of transcriptional control. So how we're controlling how transcription happens in the cell. Now these can be done through repressors that turn genes off or activators that turn genes on and actually make them transcribe more. And a lot of this was discovered through experimentation with the bacteria E. coli. Now E. coli can be good or bad. There's the E. coli that makes you sick and the E. coli that lives in your gut. But what they did is they would do a lot of experiments with E. coli and exposing it to different growth media and then seeing how the actual E. coli reacted. Now, E. coli genes, of course, are prokaryotic genes because E. coli is bacteria. Bacteria are prokaryotes. And they have something special called operons. Now, operons are a set of genes transcribed in a single mRNA. So they might be genes that are very close together, and they'll all be transcribed at once. Now, eukaryotic cells really don't have operons. Our genes are regulated individually, which we'll talk about later. So let's take some examples of these operons that are common in E. coli and how those genes are regulated. So the first example that we'll be looking at is the tryptophan operon. Now remember, the operon is a set of genes within one region. Now eukaryotic organisms do not have operons. Their genes are regulated individually, but it's still a great example to look at gene regulation and gene expression. All right, so E. coli has to synthesize tryptophan, and it does this from enzymes. And these enzymes are created, because enzymes are proteins, from different genes. And there's 5 genes within this operon that will create the enzymes that need to synthesize or make tryptophan. Now, when the cell or E. coli has enough tryptophan, say the E. coli in your gut has just been exposed to a large meal that the person ate, it can turn this gene off because it doesn't need to make any more tryptophan. And it does this through a special part of the gene. Now, of course, this gene has a promoter and then these structural genes to create the enzymes it needs. But when there's adequate tryptophan, we will actually have tryptophan acting as a co-repressor on this special repressor molecule. And this is going to attach to the operator of the gene. And this is the section that this repressor attaches to so that RNA polymerase cannot go down the gene and end up doing transcription. So let's take a look. When we have adequate tryptophan molecules in the body, this tryptophan will then attach to our repressor. And the repressor will attach to the operator, and the operator is here. So now, when the RNA polymerase binds to the promoter, it can no longer complete transcription, and then we do not have the transcriptions of these genes. So again, this tryptophan is acting as a co-repressor. It's acting to create a conformational change on the actual repressor so that it can bind on the operator. And then as the RNA polymerase tries to initiate transcription, it cannot because the repressor is blocking it. Whoops. So that's the tryptophan operon. It's an example of a repressible operon. Now let's take a look at the lac operon. Of course, you've all heard of lactose, probably in the context of milk. Now, the lac operon is a little bit more interesting because it has both a repressor and an activator. Now, E. coli are going to generally break down glucose for energy, but when it needs to break down lactose into glucose and galactose, they're going to need several different enzymes. So these are the 3 enzymes that they need. No need to memorize their names, but know that there are 3 different genes that code for these 3 different enzymes. And here they are. Now, naturally, there's a lac repressor on this particular operon, and it's going to keep these genes off when there's no lactose present, because why would we need to break down lactose if there's no lactose present? So we don't even need these particular genes to synthesize those enzymes, or to go on and transcribe and translate those enzymes. But when we consume things like milk and then the E. coli is exposed to certain types of sugars, allolactose is an isomer of lactose. What it's going to do is it will bind to, here's my allolactose, okay? My allolactose is going to bind to my repressor, there you go, and then the repressor will leave our particular operon, and then we can actually have an easier time for RNA polymerase to attach to the to attach to the promoter and then continue transcription. Now, there's a second part of it as well. This also needs an activator. Now, when lactose and glucose are both present, the E. coli prefers to synthesize glucose, but when there's no glucose, we have to be able to turn this gene on somehow. So what happens is the production of cyclic AMP is going to increase as our presence of glucose goes down. So the less glucose we have, the more of this we will have. And this is going to actually act as like an allosteric enzyme, and it's going to attach to our CAP, or our Catabolite activator protein. Now this special protein is an activator, and so here we go. This now can then bind to our operator. Oh no, we are missing our cyclic AMP. Oh, here it is. Okay, yes. It's going to stimulate our gene function. So now that the cyclic AMP is attached to our CAP, this makes it easier for the RNA polymerase to actually bind, and it's not going to bind naturally very well. So it needs the CAP here to do a better job so that the RNA polymerase can begin transcription. So it increases affinity for RNA polymerase to the actual part of the promoter, and then this is an example of positive gene regulation. So this is going to increase gene activity because of this molecule here. All right, I think you got it? If not, go back, watch again, and thanks for watching other operons.